Now, non-small cell lung cancer is the largest tumor type globally by mortality. 2 million people die every year of non-small cell lung cancer globally, but also by annual sales, 34 billion in annual sales last year. Now, our drug goes well beyond non-small cell lung cancer. It works in any cell that is telomerase positive. And 85% of cancer cells are telomerase positive in aggregate. Some cancer types can be 100%, like small cell lung cancer. Others are 85%, 90%, like liver or colorectal or non-small cell lung cancer. And our data in the preclinical setting was so convincing that when FDA awarded THIO 3 orphan drug designations out of the first 3 applications in hepatocellular carcinoma, which is the dominant histology in liver cancers, about 85%, in small cell lung cancer, the deadliest form of lung cancer, and in brain cancers, including malignant gliomas. So now we're putting forward plans to expand to these additional cancer indications, and we expect trials to start there as well in the course of next year.
Very good. And so, I mean, with the orphan drug designations that you've received, the FDA must see some real promise behind that, right? I mean, this is not, something that's easily given. How is it that your lead compound differs from other cancer treatments that are currently available?
Well, first, it has a very unique mechanism of action. So imagine the chromosome, usually represented as an X, that contains our DNA, our genetic information. And at the end of the chromosome arms are the telomeres, our target. The telomeres are also DNA sequences that are, in essence, protective end caps that protect the integrity of the chromosome in the course of the cell division cycle. The cell divisions are always imperfect, so for the chromosome material to translate in full to the next daughter cell, the telomeres will take the hits. They are built and maintained by the enzyme telomerase. Telomerase is present in all normal cells in the first year of life. But at about age 1, telomerase goes away. The telomeres have reached their maximum length, and from that point on, with each cell division cycle, they lose a little, until in the old age they become critically short, can no longer protect the chromosome, mutations begin to appear, and with this, diseases of the old age, including cancer. Now, in cancer cells, something extraordinary happens. The enzyme telomerase is turned back on. And so the cancer cells regain their ability to elongate their telomeres, and so reach a state of replicative immortality, meaning they continue to divide and the cancer grows in the otherwise aging body. And this is where THIO comes in. THIO, which is short for 6-thio-2'-deoxyguanosine, is a guanine analog. It's picked up, it's one of the building blocks of the telomeres. It's picked up by telomerase instead of guanine, placed in the telomeric structure. This creates a faulty, unstable structure. The telomere collapses, the DNA unwinds, and the cancer cell dies. This process is fast and efficient. THIO directly kills these cancer cells in 24 to 72 hours and directly kills 70 to 90% of the cancer cells. Then follows its immune effect, its immunogenic effect. Of the telomeric fragments, micronuclei are formed that present these fragments to the immune cells, becoming neoantigens in the immune pathways and triggering an immune response so efficient that if you follow THIO with an immune therapy, such as a checkpoint inhibitor, you see complete response, no recurrence, and anti-tumor immune memory. Cure. And we have seen this in preclinical setting in multiple tumor types, in lung cancer, in liver, colorectal, small cell lung cancer, brain, with THIO in combination with several different checkpoint inhibitors, Libtayo of Regeneron, Keytruda of Merck, Tecentriq of Genentech, Opdivo of Bristol Myers, not Opdivo, yes, Opdivo also. And the first go-to-market is with a clinical supply agreement with Regeneron for non-small cell lung cancer. That's our market entry strategy.
Very, very good. And you have to have faced, quite some significant challenges as you've gone through things. What are they, and how have you overcome some of the greater challenges that you found?
Yes, so I would say the most important dimension is financing. We finance the companies successfully given market conditions along the way. We started out in markets that were good and then turned super hot, in which financing for a small company was not easy because all the attention was on the bigger companies. And then the markets turned down and financing simply became unavailable. So then it was even more difficult from a different angle. But we are in a very good spot right now, and have always managed to move the business forward. I think we moved very fast to get to this point in the clinic in such a short time interval. And we managed to find a very good partner in Regeneron with a clinical supply agreement. We hope to add more partners to the lineup in the near future and go commercial in a couple of years.
Perfect. And I mean, it's always interesting to me to see the different ways that people approach the financing challenge, because I do feel like, more companies are facing this today than ever before. And to have, a company with promising information that can continue to come out, obviously it makes things a lot more beneficial whenever investors look at it and say, hey, look, this company is actually making significant progress as well. And so, it's so important. And I think, all the things that we've heard today about MAIA, that's got to be one of the things that I'm hearing as well, is that, just the significance and the amount of news that you would have as well throughout time. So congratulations on that. How does your company approach the balance between scientific innovation and commercial viability?
So you have to. In the part of it, since scientific innovation is, I think, critical, because that's where it all starts. So we got to have a compound, ideally, that hits a target that no one else does, like THIO. Has a telomere-targeting effect, and also partners well with other compounds, because in oncology, oftentimes treatments are made of 2 or 3 doublets, triplets of therapies, different therapies that target cancer from different angles. So if the drug is not safe, has some significant toxicities, it's not combinable easily. So that's key. And we have that as well here. Very combinable, toxicities are very, very minimal. Then you need to go to tumor types that have significant potential and competition is limited. Our market entry strategy is going to be non-small cell lung cancer in third line of therapy. Third line of therapy, non-small cell lung cancer in patients who have become resistant to checkpoint inhibitors and to chemotherapy in previous lines of therapy. This is now the largest unmet medical need segment in non-small cell lung cancer. And no other company is targeting, nothing is approved here, nothing is targeting this, it's free of competition. It's an excellent place to address. These patients are in a desperate clinical scenario. Unfortunately, their survival is less than 6 months. And we are showing that we improve that by a lot.